Good afternoon, everybody, and welcome to this, our fourth Tree Council seed gathering workshop of the autumn. I'll say hello properly when you're all here. We've got people flooding through the doors at the minute, so we'll just give it a second. As is the way with most webinars and things, it's always good to know where you are. So in the chat, if you would like to just say where you are in the country, just so that we get a sense of where people are listening to us from, that's good fun. And if it's a different country, it's even more interesting. So if you could just use the chat to tell us where you're all from, it just gives us that minute or two while people arrive. So we've got Jane in Salisbury. Hello, Jane in Salisbury. We've got Laura in Eastbourne, Sue in Shrewsbury, and Jim in York, and Geraldine in Hackney, and Bo in Somerset, and Owen in North Yorkshire. So they're all over the country, Ted. Just there's Diane in Somerset, and Flintshire, and Burgess Hill, and Bath, and a whole range of Congleton. We haven't got abroad yet, but we're fairly widespread from folk all over the countries. There are a few more still to come, folks, so we're just going to give it another minute, and then I will formally start, and we will go. So few rules of engagement. If you want to ask Ted a question as we go through the webinar, please put it in the Q&A. If you just want to have, and we will do our best to come back to those with Ted at the end of the session and try and get answers to as many as we can. If you just want to converse about what Ted said, but it isn't a question, then please use the chat. Jan, no problem if you have to leave at 1:30. we understand life happens like that. Mike Gardner from Dorset. Hello, Mike. So one more minute, just let the last few come through the doors, and then we are going to start. Right, I think we will begin. So good afternoon, everybody, and welcome to this. This is the fourth Tree Council seed gathering event this autumn, looking at the fascinating world of trees and seed. And I'm very, very delighted this afternoon to be joined by our special guest, who's Ted Chapman. And Ted is the UK Conservation Partnerships Coordinator at the Millennium Seed Bank. And Ted is going to introduce us to the wonders of the seed bank and its vital work in both research, conservation, and tree production. And obviously, for those of you who don't know, I'm sure Ted will tell you, it's a very beautiful place because it's based in East Sussex.
West Sussex, just
Sussex. Very beautiful place. And Ted's going to tell you all about it. So thank you, Ted, and over to you.
Thanks, John. Hi, everybody. It's a pleasure to be talking to you today about our work at the Millennium Seed Bank. We're so proud of our seed bank, so it's always a joy to show it off a little bit. So the first thing I'm going to do is share my presentation with you. And John, no doubt, will shout.
That looks good.
If it doesn't work. So as John said, my job title is the UK Conservation Partnerships Coordinator at the Millennium Seed Bank. And what that means is I coordinate the UK programme of the Millennium Seed Bank Partnership. And this means I'm based at this.
Sorry, Ted, I'm just going to say to Nicola, yes, this presentation, well, the recording of the presentation is available afterwards for you to be able to watch. Sorry, Ted.
Sure, no worries. So I'm going to try and do two almost impossible things. I'm going to talk about the work of the MSB and our partnership, particularly in relation to our work in the UK and with UK trees. And because our kind of bread and butter business is seed collecting, I thought it would be interesting just to talk about how we go about making high quality seed collections for our seed bank, particularly from trees. I could spend 2 days over all of that, but I'm going to try and accomplish it all in 30 minutes. So apologies if it seems I'm rather brief. But I'm based at this building in the beautiful High Weald of Sussex. It is the Millennium Seed Bank, and it's the centre of our international network of seed collecting partners in 190 countries and territories around the world. So we're the kind of centre of a global initiative. And we are proud to be the world's largest wild seed bank. So we're in the business of banking native wild species, as opposed to many of the other larger seed banks which deal predominantly with crops or cultivated species. We deal with wild species. And we currently hold over 98,000 individual seed collections from around the world, representing more than 42, nearly 43,000 individual taxa, or 2.4 billion individual seed. So we think that makes us the most biodiverse spot on the planet in terms of species density. And those seed are stored at 15% relative humidity, so very dry, and at -20 in cold stores beneath that building you can see in the slide there. So it's an international endeavour, but we're particularly well represented in terms of the UK flora. So we hold over 8,700 UK collections, and they represent 76% of our native and archaeophyte flora. Native being the species that got here under their own steam, and archaeophyte species being those that were introduced by people prior to 1500, just in case anyone's not familiar with that terminology. And that represents 85% of our UK threatened species. So these are comprehensive collections. They're not absolutely comprehensive, as you can see. And there are a number of, we would call them exceptional species that we don't hold in the seed bank currently. So they may be recalcitrant species, things like oak or chestnut, which we can't dry and store using conventional methodologies here, or those that don't reliably set seed in the UK, for example. So there are various reasons why we haven't got to 100% yet, though perhaps one day we will, through new technologies and continued effort over the years. So that's really the seed bank as a whole. But my work is within our UK programme, and we're a team of 7 based in a building at Wakehurst. And we kind of have 3 purposes, if you like, as a team. And one is to increase the ex situ conservation of the UK's flora at the MSB. So that's the conservation of the species diversity, but also the genetic diversity of the UK flora, and hold those as seed collections in our seed bank. And crucially, to use those seed collections and all the science and technical expertise that Kew holds around the use of seed to support in situ, so in-field conservation and restoration work. And we largely do that really through the third part of our objectives, which is to share knowledge and expertise through partnership working, training, technical support, applied research. And I'm going to provide a few examples of how we're doing that with our tree seed collections and data in just a second. My background actually was in native plant production and grassland restoration. So that's why I've got a few slides of us working in chalk grasslands. But more latterly, my work's come to focus much more on trees, both the collecting of tree seed and the use of those seed in research and afforestation projects. And really, I kind of stand on the shoulders of giants here, in that Kew has been banking tree seed for many, many years. And we actually hold collections of box, I think, from the 1940s in our seed bank, which are still viable. But really, there was a sea change in our tree seed collecting work through the UK National Tree Seed Project, which some of you may be aware of or indeed contributed to through seed collecting work. And the purpose of this project, which ran from 2013 to 20, was to establish for the first time an accessible, genetically representative national seed collection of our native trees and shrubs, to conduct research to understand and overcome constraints to their conservation, ex situ in the seed bank, and their use in fields. So thinking particularly around questions of seed storage, seed processing, germination and establishment, and also some genetic questions of relevance to our work, and to raise public awareness generally around the role of seed and trees in meeting UK conservation targets. So this was a highly successful project, and we set 961 collecting targets. So our target was to collect, to make at least one collection of every native species within every Forestry Commission seed zone in which that species naturally occurs. So we mapped the species range across the Forestry Commission seed zones, which you can see in the bottom corner of the slide there. And we sought to make a collection from each one of these above and below 300 meters. So we had an altitudinal difference there, as well as a sort of biogeographic difference. This was how we sought to make our collections comprehensive. Now, we're obviously using biogeographic regions here as a proxy for genetic diversity. This is a sort of practicable way of going about organizing a collecting project, and realized 961 collecting targets. It's an ambitious number, but 702 of those were fully met within the lifetime of this project. And they represent 13 million good seeds, so 30 million viable, healthy seeds, representing almost 11,000 individual trees. And we took an unusual approach with this project. Typically in a seed bank, we collect from multiple individuals within a population to create one seed collection that's representative of that wild population. But for trees, we now collect individual maternal trees and we hold them separately. So it's a kind of highly high-grained, a fine-grained approach, I should rather say, to collecting seed. But it means that our collections can be used to for example, to study differences between individual trees within populations, as well as differences between populations and species. And also where individual trees have a particularly useful trait, for example, tolerance to a disease like ash dieback. We have the seed from that individual maternal tree available for research and use. So we rolled out this sampling strategy across 76 species, of which 50 are fully sampled. So we hold collections from all of those seed zones. Alongside the seed, we have all the field processing and germination data for each collection. The data for us is almost as important an asset as the seed itself. It tells us so much about the place that it's come from, and what we need to know to turn that seed back into plants. And we're indebted to 30 or more than 30 partner organizations who actually made 80% of those collections on our behalf. So some of that work was done by Kew, but much was done by our partners around the UK. And you can see in the background of the slide there, those are seed collections banked in the -20 in parfait jars, preserving jars there, which provide an excellent hermetic seal to keep the seed dry and safe. So already these collections have been used in a number of scientific research projects, leading to a number of interesting scientific publications, some of which are Kew-led and some of which are researchers in other organizations, including around the world. I'm going to talk a little bit more about some more sort of practical uses of the seed collections in a second. But just to say that we haven't stopped tree seed collecting. So after a brief hiatus for the coronavirus pandemic in 2020-21, we picked up work with a new seed collecting project, which is the grandly titled Diversity, Adaptation and Use Next Generation Seed Collecting in the UK. And I put in next generation because already with this biogeographic sampling strategy, so making multiple collections from across the species range in the UK, that was already a kind of novel approach for the MSB. But I want to push that further in this project. So we're making very targeted collections, looking at specific traits. So ash dieback disease tolerant individuals, for example, we're identifying and making seed collections from those. We're looking at areas of known high genetic diversity and local adaptation, and that is increasingly informed by genomic studies. So we're following the science in developing our sampling strategies. Of course, plugging those remaining biogeographic gaps, so the collections we weren't able to make through the Tree Seed project. And increasingly, we're tying our work to the species and populations that are in demand for research or restoration or reforestation use, and close links with government and non-governmental organizations really helps us tailor our seed collecting to make sure it's of greatest use. But there are a few projects that we're involved in making use of the collections directly, and they're just neat examples, I think, of what you can do with high quality seed collections and high quality data. So we're, for example, working with Forest Research and a really interesting project to establish new experimental woodlands around the UK. So these are long-term experiments that can bring together native trees of different UK provenances in effectively large common garden experiments and look at how they perform over time and through a changing climate using different metrics, some of which are biodiversity related and some of which are relevant to timber production. So we've provided population data, so where are really nice locally native large populations that can provide seed material for this. And indeed, we're providing seed material directly from our seed bank to support the development of those woodlands. We've provided a lot of population data to help identify new registered seed stands within the government seed sourcing grant scheme. So this is looking at large populations which are locally native, or we believe are locally native, have been present for a long time, that are potential sources for seed material for tree production. And indeed, we've also provided some seed to support the development of new seed orchards to provide seed material. So this is all really with an aim to boost the availability and diversity of native tree planting material in the UK. And we're also working on a new tree seed database. We hold so much data and so much of it is of relevance to research and tree production, thinking about things like seed mass, seed dormancy, seed germination, the location of and distribution of tree populations. And we want to make all of that available through a new integrated open access database, which we're working on currently, supported again by DEFRA. So there's really a lot we're already doing, I think, with the tree seed collections that were made, and indeed there's plenty more we can be doing. And then a final project that I'm leading that it's quite nice to flag up, thinking particularly about tree production, is our Seed Traits for Tree Production project, funded through the Tree Production Innovation Fund, which looks to tie research and tree production with commercial partners together. So we're actually using our seed collections to look at how seed morphological traits, so that's external characteristics of the seed, like their shape, the seed coat color, the seed mass, how those sorts of traits link to their germination, how germination responds to temperature, so what are the minimum, optimum and maximum temperatures for germination in UK tree seeds, and we can model that against climate change to see which populations and species are potentially going to be at risk from a warming climate at the germination stage. And we're working with our partners at Elsams to use this information to calibrate and test what to me seems like miraculous high throughput optical screening equipment to actually take out non-viable seed and therefore increase the quality and germinability of tree seed batches that are entering the production chain at high volumes. So all of that is really to support climate smart seed sourcing, so we can make more informed decisions about where we source our seed from to ensure it's resilient and able to regenerate under future climate situations. There's quite a lot of relevance there for species-specific pre-treatments or priming protocols potentially. The more we understand about the germination requirements, the more interventions we're able to make to improve germination and make more material available. And as already mentioned, through screening, improve seed quality and germinability, both of which are potentially significant issues in tree production and can cause significant waste also. But all of that work really depends on having really high quality seed collections and data. And all of that really depends on the way we go about making our tree seed collections. So as that's really the theme of this run of talks, I thought it'd be interesting to talk about how we go about making our seed collections for the MSB. So some of the fundamental principles of making a tree seed collection which is legal and safe, that is capturing the maximum genetic diversity. So we always want our seed collections in the bank to have captured as much of the genetic diversity of the wild population as we possibly can. So we're deliberately trying to avoid selection or selective pressures in the way we go about collecting our seed. We believe that genetic diversity really is the key to the adaptability and the potential of our seed collections to provide all the traits that future populations will need: drought tolerance, disease tolerance, ability to cope with a range of conditions. So if we can capture as much genetic diversity as possible, that's really, really important. And of course, we also want our genetically diverse collections to be of high quality and viable. So that's important right from the start. So I'm going to sort of gallop through how we go about doing that. And along the way, I will signpost further sources of advice, technical support, and where you can access data about our seed collections, if that's of interest to you. And I flagged straight up here our UK National Tree Seed Project Seed Collecting Manual, which really covers everything that I'm going to tell you now and is available to download through our website, and the link is there. So before we go into the field, of course, we want to select the sites from which we're going to collect our seed. For our purposes, we're looking for naturally occurring autochthonous, so locally native populations. So these are populations that, to the best of our knowledge, have generated naturally and have generated naturally over time. They haven't been planted by people. So we avoid plantations, and we're looking wherever possible for large populations, larger populations tending to have a greater genetic diversity within them. Large connected populations that have been able to share genetic information and maintain it. So what that in practice means in a UK context is that most of our collecting work is done in ancient semi-natural woodlands, protected areas, and other sites where we have a long management history that can make us reasonably confident that those conditions of naturally occurring autochthonous populations can be met. And species distribution and habitat mapping can be helpful. And of course, we're blessed in the UK that our flora is extremely well mapped and understood by international standards. But local contacts and expertise, of course, are really invaluable to lead us to really high quality populations and high quality collecting sites. Of course, our collections need to be made legally and safely. So we, for the most part, are not marketing our seed material. So we're not usually too bogged down with guidance and regulation around the marketing of forest genetic resources. But we do for sure have to obtain landowner consents for the seed collections that we make. And additional consents are required, of course, for collecting on sites of special scientific interest. So all of that work has to be done beforehand. And you can see on the screen there, that's our standard landowner consent form, which we use and which covers the future uses of those seed collections and data. So we know we're free to distribute and use that material as we would like to. Of course, we'll conduct site-specific risk assessments. We have to have proper insurance. These are important considerations when making seed collections. And we will also consider biosecurity risks, of course. So some species are quarantined. Very few UK tree species are currently subject to a plant health order. But we will consider risks. For example, with juniper, it's not a quarantined species, but there are emerging risks around Phytophthora. So we will put in additional regulations to make sure that we're not transferring Phytophthora from site to site or from population to population within our seed bank. And we'll mitigate any risks we identify at that stage. And there's more information about biosecurity risk assessment and also the kits, the disinfection kits that we take into the field in the collecting manual. So we're careful to avoid spreading plant diseases around through our work. And then we finally make it into the field. And of course, we want to be sure we're collecting the right species. That seems straightforward enough, but actually can be extremely complex, particularly for species which readily hybridize, like some of the willows, for example. The identification is actually a specialist skill. We will assess and record the size and extent of the population. The number of individuals within the population is extremely important to us. It's really key data to understand the potential diversity of the population, and also how successfully we've captured that through the number of trees we sample from. So we will always make an attempt to assess the size of the population, though it can be very difficult, of course, to do that accurately. And we'll always cut test 10 or 20 seeds. So we'll cut them open and look at them with a hand lens, usually in the field. You can see some examples there in ash. And we're trying to assess a couple of things there. So we're looking at how mature the seed is. So is the seed material ripe and therefore likely to be viable and also have the longest longevity in storage? So we're always aiming to collect at or very close to the point of natural seed dispersal. And also we want to assess seed quality. So we want to make sure that the seed is full and healthy. So on the right-hand side of that top picture there, you can see a nice white filled seed, which we would deem healthy compared to the other seed on the left, which is clearly kind of brown, not full. It looks a bit necrotic and dead. So that can save us wasting time collecting seed which is not viable, and can also help us collect enough seed to make sure that we've accounted for the proportion which is not viable. And typically in wild seed populations, there'll always be a fraction of seed which is not viable, and that can vary from place to place and year to year. This enables us to estimate the quantity of mature, healthy seed in the population. And that's important because we aim always not to collect any more than 5% of the seed which is available on the day. So we don't want to strip populations of their seed. That's how they're reproducing. We need to be careful that we're not interfering with their ability to reproduce. So we need to know how much mature seed there is, so we can only take a small fraction of it. So we will aim to collect randomly and evenly across a tree population, and we will avoid selection. Of course, if you're making a seed collection for the purposes of selecting useful traits, you'd approach this very differently, but we don't want to make that selection. So we will collect randomly and evenly across a population, usually aiming to collect from trees that are at least 50 meters apart. We're going to collect from all parts of the tree because pollen genetic information may be arriving from different directions. So we want to collect from all parts of the tree. And we aim to collect 15 or more trees per population. Of course, the more trees we can collect from, the better, but there's always going to be a trade-off between the diversity of your collection and the time available in the field to make it. So having reviewed the available scientific evidence and literature, we aim to collect 15 or more trees per population. Generally speaking, as I said, we collect individual maternal lines, but sometimes we may combine multiple maternal trees into a single population collection. It really depends on the purpose of the seed collection, but because our seed collections are partly for research use, future research use, we hold them at maternal line. And we collect what you need for your purpose without collecting unsustainably. So there's no point collecting more seed than you need. A really nice conservation collection for us is 10,000 or more seeds. So that's small, very, very small by commercial standards, but actually represents quite a lot of collecting effort, bearing in mind we're trying to get it from at least 15 individuals across a population. So there's always a bit of thought that needs to go into the sampling strategy. The collecting itself, we mostly do that from the ground using long-handled pruners or throw lines and shaking. Sometimes, of course, we'll use climbers to go up trees and make collections on our behalf. That's obviously a skilled job, but sometimes that's the only way of doing it. And alongside the collections, of course, the data is crucially important, particularly for us. A lot of the research value stems from the context that the seed collections come from. So as an absolute minimum, we need to know where they've come from. We need to know who owned the land. It's extremely useful to know what else was growing with the species to give a snapshot of that ecological conditions at the collecting site. And of course, we gain data all of the time through the work we do processing those seed collections and germinating them. We'll also routinely take an herbarium specimen, which you can see at the top of the slide there. That's a pressed, dried part of the tree that we've collected from. That enables our experts in herbarium to verify that the tree is what we think it is, and of course is a lasting biological record in its own right, and actually are increasingly used for things like morphological studies and also DNA extraction. So these herbarium specimens are valuable in ways that we actually hadn't understood when we started taking them. So that's important for us, for sure. And then thinking about seed quality. So there are some basic principles behind making a really high quality seed collection in terms of the viability of the seed. And the first really is to collect seed at the point of natural dispersal. So this is really when seed is at its most viable, and also has the greatest tolerance of desiccation. So the greatest ability to dry and remain viable and healthy at low moisture content. So that for us, of course, as a seed bank is extremely important. So we always aim to collect at the point of natural dispersal, and where we collect immature seed, or sometimes we have to unavoidably, we will ripen it under ambient conditions before we dry it down. So we allow it to continue ripening before we halt those metabolic processes through drying, or slow those metabolic processes through drying. And having collected seed at the point of natural dispersal, we'll then seek to keep it dry and cool. And we're kind of guided in seed conservation by Harrington's rule of thumb, which states that the longevity, the lifespan of seed doubles for every 1% reduction in water content or 6 degrees centigrade decrease in temperature. So when we dry our seed collections from ambient conditions, which may be 80 or more RH in the UK down to 15%, the seed moisture content will also reduce. And with every 1%, we're doubling its seed lifespan. So you can see in that reduction from ambient to 15%, we're getting doubling many times over. And then by storing our seed at -20, of course, we add in the temperature effect. And that enables us to extend the lifespan of our seed collections to decades or centuries through conventional storage. Of course, we want to lock that in at this collecting point of collection. So we'll always collect into porous bags so the seed can breathe and start drying right from the point of collection. Selecting nice dry seed at the point of natural dispersal. Again, you can see we did a lot of collecting work in ash, which provided a lot of really nice pictures for presentations like this. But of course, we also collect fleshy fruits. So in the case of fleshy fruits, we'll usually seek to collect into plastic bags in the first instance, but then we'll want to get that flesh off as quickly as we can so that we can start drying the seed and start locking in the benefits of drying for the longevity of the seed. All of this is covered by our technical information sheet 3, which is also available by via our website. So we'll be monitoring how the seed collections are being dried. We'll do this under lab conditions within the MSB, but this can also be done under open barn conditions like this, using handheld hygrometers or data loggers with hygrometers and temperature sensors. So these are low cost, low tech pieces of kit that can really help guide how you're drying seed collections down after harvest and really maximizing their quality in the longer term. And again, we've got some technical information sheets which cover this, both at the technical lab-based end and the less technical, more sort of homemade approaches, if you like, to seed drying and conservation. So for example, we've developed these blue drums you can see on the left-hand side there. These are sealed drums with silica gel. Similar devices have been created using dried rice and charcoal and other substrates that can basically reduce humidity within the space and support the drying and short or medium term storage of seed. And some of our partners also use these dryer incubators you can see on the right hand side here, which are sort of fridge sized pieces of kit, which which can be humidity and temperature controlled. If you don't have access to a large cold store, as we're lucky to have at the Millennium Seed Bank. So I've mentioned that all of what I've told you now and much more is publicly, openly available. Our tree seed collecting manual covers the in-field techniques of all the planning and sampling considerations, and then the MSB's technical information sheets cover everything else in considerable detail. So if this is of interest to you in your work, do please take a look. And if you'd like to take a kind of closer look at what we hold in terms of tree seed collections, this data is mapped with some sort of headline information around population size, for example, sample size, the number of seed we hold, etc., via the UK Forest Genetic Resources Interactive Map. And that's available online, and you can search by species or look at everything all at once if you like. So that's quite an easy way to just access and just have a little look at what we already hold in our seed bank. And that's everything from me. Thanks very much for listening, and I'll be happy to take any questions.
Thank you, Ted. So if you could just take the presentation down, thank you, and then we'll have a we'll tackle some of the questions that people have sent in. If you have any others, please do pose them to Ted as we go through the remainder of the session. One from me to start with. You said that seed can last in a seed bank for a fairly long time. You said potentially decades or centuries. Is that all seed, or
no?
Every tree species, or do we know what the shelf life of some of them are?
So there are some. So there is a spectrum of seed storage behavior. Is probably the best way to describe it. From those species which are utterly recalcitrant, so species like oak, which we cannot store under these conditions. There are techniques to store these species under cryopreservation, and we do that at the Millennium Seed Bank, usually as excised embryos, which are then frozen at very, very low temperatures, through to species with very long with very long longevity under dry cold storage, which can be measured in the centuries, and then there's everything in between. So thinking of UK trees, we can't bank oak or chestnut. Species like the willow species are short-lived. Hazel is what we might think of as an intermediate species. We can store it, but under conventional storage, the lifespan is measured in a short number of decades, through to most other species where we can't, in the decades that we've held tree seed collections, we can't really see significant decline in viability yet, which actually makes it quite hard to say how long they will last, because we haven't yet got enough data of decline to model it accurately. But we are there for most UK species. We're measuring lifespan in the seed bank in decades or centuries. Yeah.
Megan actually asked, what's the oldest seed that you've got stored at the seed bank currently?
Well, I know the oldest tree seed collection we have from the UK is from the 1940s. We hold non-woody seed collections from the UK from the 1930s. I don't know what we hold that's older than that and is still viable. But for sure, some of our oldest collections are pushing 100 years now and still have viability. But the vast majority of our collections have been made since the Millennium Seed Bank was established in 2000, of course. And that's why we don't have enough data on viability decline yet. Yeah.
No, fair enough. The sort of maxim that used to be spoken about, when I started in this line of work, was that in an ancient woodland, if the trees were cleared, some of the seed might last anything up to 100 years just in the soil. Do you know whether that was just apocryphal, or whether there's any science behind the length of time that seed can be stored not in seed banks?
So sort of seeds, the longevity of seed in the soil seed bank is a fascinating area of study. It's not something we do a great deal of at the Millennium Seed Bank, actually, because of course our conservation is ex situ. Um, I would imagine that the proportion of seed that is viable obviously is going to decline over time, and the speed of that decline will relate to the conditions that the seed is in. So tree species sort of regenerating spontaneously, the likelihood of that's going to decline over time. I don't know what the top limit for that is for any given species. I would think hundreds of years is probably apocryphal, but I stand to be corrected on that.
An interesting one to do a bit of.
Yeah.
Jane and Nicola both asked questions about elm trees and whether you know anything about disease resistance in elm trees through their seed, or whether the seed conveys any element of resistance.
Okay, so it's an interesting question. So we're doing a little bit of work with Natural England at the moment, actually, around elm subspecies or species which appear to be resistant to Dutch elm disease. Now, there's some taxonomic debate about whether these are truly individual species, subspecies, or just within the English elm population. But there do appear to be some individuals that are resistant to Dutch elm disease. And so we've been collecting from these individuals, propagating them at Kew with a view to establishing them at different Natural England managed sites to expose them to disease and see if they are indeed resistant. And there is also some work being done around resistance in English elm populations. The heritability of this trait through seed is not something I think is fully understood yet. So the question is whether we can breed, we can isolate this trait and breed it into resistant elm populations, and whether we can do that through cross-pollination or through vegetative propagation from tolerant individuals, I don't know. But there seems to be some resurgent interest in work in this area. And certainly one of our collecting priorities is disease-tolerant elm. So if we can collect seed from disease-tolerant individuals, or putatively disease-tolerant individuals, the material is available for this research to continue.
Yes.
And that's the same approach we've taken to ash.
I guess the request then from you to anybody, Ted, is if you know of an elm tree that's looking healthy, it might be something that's worth flagging with your local authorities or even seed bank.
Yes, a really healthy elm, particularly if it's not a hybrid elm that's been planted, and you're confident there's lots of Dutch elm disease all around. So it's surviving because it has some tolerance characteristic rather than it's just sort of the only elm within 50 miles. Those are the really important individuals. I think it's worth flagging. Yeah.
Okay. So Jackie says, does the viability of tree seed vary with the age of the tree from which it's been collected? So does the age of the tree affect seed viability?
It might, if the vigor of the tree was affected by its age. So viability is impacted by lots and lots of things at the maternal stage whilst it's attached to the mother plant. And viability can be undermined if the mother plant is under stress. And that stress may be imposed by age, may be imposed by environmental conditions or disease, or a combination of all three. So we will collect from all age classes within a population because that's important in representing the population in our seed collection. But we do see in our maternal level collections, now we can compare individuals within a population, that viability does vary considerably between individuals. Unpicking the reason for that is fiendishly difficult because it may relate to the health, the age, the location of that individual within the stand, how much sun it had, drought stresses, pollination, how effective pollination was. There are all sorts of factors that play into that. Age, I suspect, is one of them, but we don't avoid collecting from old individuals. but we will always do that cut test to make that initial assessment of seed health. Yeah.
One of the things that I don't think everybody always understands is that trees need to become sexually mature before they produce seed. So that element of hawthorn may be taking 7 years to get to a point where it flowers and produces seed, but an oak tree maybe takes 40 years to get to a point where it's producing decent seed. It's just a point that I think not everybody gets, that the tree has to grow up a bit before it starts to produce lots of seed.
Yes, indeed, that's right. And of course, some trees require male and female trees to be within pollinating distance. So that can be a real issue for things like, rare species like black poplar. That can almost be an existential issue where you have a lonely male or a lonely female, and the individual may be terribly healthy, but there'll never be any seed without some intervention. So understanding the kind of reproductive biology, if you like, of the trees is quite important. Yeah.
Claire's asked a question: when and why might you take seed out of the seed bank?
Oh, so the seed, it is a bank. I like to think of it as a bank in the sense that the seed is going in, but it's also coming out. So the seed is available for request through the MSB seed list. So you can request seed samples for research, conservation, restoration, or education use. So the seed is used in things like to provide samples for genetic studies, for looking at phenotypic differences, so differences in the kind of shape and function of tree seedlings or seedling establishment. It's used, as I've mentioned, it's being used to establish seed orchards. So those are sort of, orchards that are going to produce tree seed in the future. It's sometimes, occasionally, we will supply seed for direct reintroduction purposes, usually for threatened species, of course. So the seed is going out all of the time. And part of the reason we need to keep an active collecting program is to keep making sure our seed collections are providing the material that is needed, and also that we are replacing the seed that's going out. Yeah.
And there was another question from, where was the other question linked to that same point, which was, are there times when you might take seed out and replace it with new seed? You might be worried about it's been there too long, or so do you ever replace seed? Yes.
So our standard protocol is to test seed for viability when it first enters the seed bank. So that's to assess how viable it is, and also it provides a propagation protocol. And then we retest every 10 years. And we will retest more frequently where we know or suspect a species may be short-lived. So we have found, for example, that beech and hazel collections are proving to be relatively short-lived under dry cold storage. So we now routinely bank a portion of those under cryo storage over liquid nitrogen. And we're also starting to recollect in beech and hazel, where we know we've got fast viability loss. So yes, we always have to be mindful of how the viability of the collections is declining and make new collections where we have to, or find technological solutions like cryopreservation to eke out the lifespan of the collections we hold.
So thank you. So Nicola says you should go to Leicestershire, because she's still got some very large elms in her part of the world that have survived Dutch elm disease. So there's an offer from Nicola to go to look at the trees in her part of Leicestershire. We'll come back to that later.
That's interesting.
We'll try and get details from you, Nicola, so we can pass it on to Ted. So in some years there's huge quantities of seed, and some years there isn't, and that's the masting process. Owen wants to know whether you try to collect seeds during mast years for maximum yield, or do you just collect it at any time whenever it's convenient?
Well, I don't know. We find mast years rather hard to predict. So we will the way we do it is we'll try and we'll have a list of targets that we'll aim to collect over maybe a 3 or 5 year period. And in any given year, we may seek to collect ash, for example. So ash has been a priority for collecting disease-tolerant ash. Ash took 2 years off. It had a huge mast year in 2020, and in 2021-22, it produced virtually no seed. So we had intended to collect seed in those years, and we couldn't. This year there is seed, so we will collect. So we don't try to predict masting in our plans, but we try to accommodate it by having other species we can collect from if the target has taken a year off. Obviously, when a mast year comes along, that's helpful to us because it does increase yield. Yeah.
Okay, thank you. There are a couple of questions that are sort of about the differences between genetic diversity and species diversity and all those sorts of things. So maybe it would just be worth, you talked about collecting different seed at different altitudes. Maybe it's just worth exploring a little bit the difference between genetic diversity and phenotypic diversity, and just a little bit of that in terms of how you select to make sure you get maximum genetic diversity.
Yes. Well, I should caveat by saying I'm not a geneticist. I'm hugely interested in genetics, and it does inform virtually everything we do. And we have a conservation genetics team at Kew to help us with that. So I suppose I have quite a working, like practical understanding of these things. But the way our sampling is structured, we usually lack a proper molecular understanding of where genetic diversity is occurring and where one population may be different from another, and where one population may be more diverse than another. So we're using the things like the biogeographic seed zones and the altitudinal distinction, and things like population size are in effect proxies for genetic diversity. So we're using these to guide us to populations which we have good reason to think are likely to be genetically distinctive because they're growing under different ecological conditions or at different altitudes, and are likely to be diverse because they contain many individuals and are able to share genetic information freely. So there's less chance of inbreeding. And we have done some studies to look at how successful this approach is. One colleague in America did it was a modeling study on ash, and our colleagues at Kew did a molecular study in yew. So they looked at the diversity within our seed collections and compared them to diversity within the wild populations. And they did find that we were capturing the vast majority of genetic diversity through this sampling strategy. So it was a good approach, but inevitably, the more rare alleles were, the more rare little adaptations were, the greater the chance of us missing them. And we know that's always going to be the case. We're always going to lose something when we make a collection. So it's all about minimizing that, really. Phenotypic differences, of course, the differences in the way the plants, so the genotypic is within the genes. Sometimes we know, but often we don't know where that's occurring. So we use these proxies. Phenotypic differences, of course, we can see, but it's very hard to know when trees look different, whether that's genetically driven or whether that's driven by the growing conditions or the way different genes have been expressed. And that's why we will consciously seek to avoid selection. So we will collect seed from big ones, little ones. Early seed, ideally we'll collect, and later seed we'll collect. So we'll try to capture the phenotypic range as well as the genetic range within any population as best we can. Does that answer it? It's a complicated thing. That's my best attempt, I think.
It does, and unfortunately, I didn't quite get it in the photograph, but just behind this is net-leaved willow. The photograph behind me, well, is what I'm talking about. For those of you wondering, he's blathering on about. In this photograph behind me, this is net-leaved willow, which is one of the high mountain willows. unfortunately, just the other side of this little lump of limestone, so you can't see it, was something called mountain birch. And mountain birch is a form of downy birch that grows on top of mountains in Scotland. And this is the absolute epitome of what Ted was just saying. Knowing whether that's a genetic, it's tiny, that has little microscopically small leaves of less than a fingernail and grows as a very small plant on a mountain. What I've never been able to find is whether that's a genetic difference that's causing that tree to be like that on the hillside, or whether that's the way the tree is growing on a mountainside. It just makes it small. It's just bonsai. That's the difference between genetic and phenotypic. And it was quite stunning standing there looking at this tiny little microscopic birch tree thinking, how on earth did that one get there?
Yeah.
We have, I think, time for one last question. So where should we go for our last question? Let's see which ones we haven't, whether there's a topic we haven't covered. It was just George asked for just for final clarification of your 5% collection rule. And he said, did you mean the 5% of the population or 5% of the individual? So 5% off one tree or 5% off all the trees in the area?
It's of the seed that's available on the day.
Yeah.
Across the population.
Okay. So that might mean you take a bit more than 5% off one tree.
Yes, although we would really try to avoid that.
Stripping it, yeah.
Yeah, we always want to avoid stripping it. It's different, obviously, if you're in a seed orchard, and indeed trees are unlike an annual species. Trees are more resilient to over harvesting in the sense that they're going to have several years in which to set seed. So it's not like one regeneration opportunity which is gone. But nonetheless, there are plenty of other organisms that depend on the seed as well as the plants themselves. So it's really important to take a responsible attitude to that, I think. Yeah.
Fantastic. And responsible attitude to seed collecting is what we're all about, folks. So don't go strip those trees out there. Ted, thank you so very much for your time. This is the last, ladies and gentlemen, of our 4 sessions this autumn. If you want to see them again, they will all be available from the Tree Council website over the next few weeks, looking at different aspects of seed and seed gathering. Thank you very much, Ted, for your presentation this afternoon. As you can see from the comments in the chat, everybody's loved it.
Great.
So thank you.
You're welcome.
Thank you very much indeed. Thank you all for joining us this afternoon. And we hope you have fun with your seed collecting. We hope you enjoy your seed collecting. We hope you grow your trees from seed. Thank you for your time. Thank you for your presence, and we'll see you all somewhere. Thank you.
Cheers.